Hello friends, you remember in first lesson I talked about the nucleic acids and the nucleic acid DNA, but you remember we said there are two types of nucleic acid. The first one is DNA, but the second one is RNA. RNA ribonucleic acid is available in the nucleus, in the cytoplasm as case in the nuclei, and in ribosomes in other structures. This acid can be genetic material for some viruses. So there are viruses which only DNA with only RNA, or some of them with DNA and RNA. Also, it's very important in protein synthesis because it assists the DNA to produce proteins and enzymes, because most of enzymes in protein structure. This acid is similar to DNA in chemical building units because both of them made up of nucleic acids, but it differs in some ways. How the RNA contains the ribose sugar. You remember we said DNA, the pentose sugar in DNA is deoxyribose, so we call it deoxyribonucleic acid, but in RNA is ribose sugar, but not deoxyribose sugar. With what? With one less oxygen. RNA contains nitrogen base, uracils, but not thymine, which we said the thymine for only DNA, but the uracil only for RNA, what the other nitrogenous bases are common. RNA usually consists of one chain but not two as DNA. DNA consists of two chains of nucleic acid which base and complementary strands, but the RNA consists of only one. But in some parts of the RNA may be bent to become binary chain, and inner made up of one chain, but if it is bended, these bends can connect by hydrogen bonds and look like two chains. In that uracil connect with adenine and cytosine connect with guanine chains. RNA is short, nearly equals the length of the one gene, because for each gene is responsible for expression of one trait, producing one specific protein. So the length of RNA is length of gene, but the DNA is a huge molecule, contains hundreds or thousands of genes as a size of chromosome. RNA carries instruction of protein synthesis because no protein synthesis takes place in ribosome, which found in cytoplasm, but DNA is found in nucleus enclosed by membrane. But the DNA can give the instructions only which is carried by RNA from nucleus to cytoplasm. RNA can behave as enzyme, but the DNA cannot do enzyme function. So in this ways, DNA and RNA different one from another, like a comparison between two kinds of nucleic acids. In function, there are three types of RNA. We will mention the three types of RNA, and all of them are made in nucleus of the cell and transferred to the cytoplasm. So it has role in protein synthesis. As we said RNA assists the DNA in protein synthesis. First one is messenger RNA, or we can show it by mRNA. It is a molecule transfer the genetic messages from DNA, which is available in the nucleus, to ribosomes, which are available in the cytoplasm of cell in eukaryotes. The ribosomal RNA, from name we can understand, is a part of structure of ribosome. It shares with protein its formation, means the rRNA plus some proteins form the ribosome, and the function of ribosome is building proteins. Transfer DNA, RNA, it transfers amino acids to build proteins to the ribosome for protein synthesis. It was noticed the amino acid connect this acid RNA before transfer it to the ribosome. They connect amino acid, carry them to the ribosome. Genetic code and the messenger RNA. Genetic code points out the succession of nitrogenous bases in messenger RNA because it directly copied from DNA. Like DNA can copy itself, but RNA cannot copy itself, but it's transcripted by what? By DNA. So 3 nearby nucleotides represent codon. So you remember when we show the nucleotides of DNA, we put them in groups of 3. That's why in messenger RNA, each of these 3 nucleotides, we call them as codon. Like each of this nucleotide, one letter, but each of these 3 group codons like a word. DNA and its ability to transcript the RNA. So we studied DNA duplicate itself, and we call it as replication. But forming RNA from DNA, that time we call it transcription. You know, there are transcript papers you put in under a white page when we write on the page above, but directly what you write pass it through the transcript to the another paper, but the color of writing is as the color of transcription paper. So transcription is the building operation of RNA by using one chain of DNA as template. Transcription is regarded as the first stage of protein synthesis. Why? Because the messenger RNA, we say, carry the message from DNA to the ribosome. So the first step is building of messenger RNA, which we call it transcription. So it is the first step. First, RNA polymerase enzyme. As you remember, for building new DNA, we call it DNA polymerase enzyme. For transcription of RNA is RNA transcription polymerase enzyme, which is the enzyme that stimulates the producing RNA from DNA template. Connects with promoter situation that leads the unit, the rapid DNA chains and their separation. So the RNA polymerase enzyme connect a piece of DNA, then the DNA from that point start to separate 2 chains of DNA, separate one from another to build RNA. RNA polymerase enzyme adds RNA-free nucleotides to nucleotides which are in one of the DNA chains, and it produces a new RNA chain. As the case in DNA replication, so the repair of the complementary base determines nucleotide succession in RNA. As example, for example, this is the template DNA with A, T, A, C, A, G, G, A, T, and A, T. So this template, what? DNA, transcripted RNA, A completed with T normally, which is thymine, but in RNA there is no thymine. Instead of thymine there is uracil, so it completed with uracil. Thymine with adenine, A with uracil, C, which is cytosine with guanine, A again with U, uracil, G with C, G with C, A with uracil, T with A with U, and again A with U, and T with A. So this is the sequence of nitrogenous bases in RNA, and each of these 3 nucleotides, we call them as what? Codon, like first codon and second and third and fourth. So the sequence of nitrogenous bases in DNA determine the sequence of nitrogenous bases in RNA. For example, Another example, if the succession of the bases in DNA is CAGCTA, so the succession of bases in RNA chain will be as follows: C completed with G, A with U, G with C, C with G, T with A, and A with U. A not completed with T, but completed with U, because we said thymine is unique for DNA and uracil for RNA. After RNA polymerase enzyme have left the area of gene which had been transcribed, the DNA chains wrap again. After RNA polymerase enzyme arrives to the ending signal, it releases new RNA with different types. The product RNA may perform its function in the cell. The enzyme can transcribe another gene because enzymes are real. So this picture we see again it about the DNA, transcripted messenger RNA. Each of these three nucleotides, we call them as what? Codons, which is messenger RNA transported to the ribosome in cytoplasm to be used in protein synthesis. And we remember the proteins are made up of smaller units which called as amino acids. So this codon AAG, like it's a code for what? For amino acid lysine. Another code GAG for another and another and another for different many amino acids. So the sequence of nucleotides determine kind of amino acids which determine kind of protein.